Hi, this is Mr. Titanium, i.e. Richard Wagner. I'm your host, Richard Wagner. I'm so happy to be here. Today, we're going to discuss the structure of DNA. And what a great time to do this, because next episode, we're going to discuss the origin of life. And then the episode after that, we're going to discuss, there was this Reddit article about alien DNA, and we're going to determine whether or not there's any truth to that particular article, or at least the genetics portion. Anyways, DNA stands for deoxyribose nucleic acid, and it all comes down to the sugar, deoxyribose. Deoxyribose and ribose, which is RNA, ribose nucleic acid, are very, very closely related. The only difference between the two is that deoxyribose, as the name infers, has lost one of its oxygen molecules, and as a result of that, it enables DNA to it enables the sugar backbone to be flexible in DNA, so that you can have the twist of the double helix. But before we start getting too involved into the structure of DNA, I want us to back up a little bit and go through some basic chemistry, so that we understand how DNA actually works. So to start with, we have to start with the first element of all. There's a bunch of elements that we're going to discuss today. Those elements are going to be hydrogen, oxygen, carbon, phosphate, and nitrogen. That's it. So this, and by the way, in my opinion, we're going to talk about, organic chemistry or molecular biology. In my opinion, those are probably some of the three most and foremost. They're the most important elements. And then, of course, there's always chlorine and other elements. Who's to say what's the most important? I guess it all depends upon your perspective. Hydrogen, number one. And it really is number one. It's number one because it only has one proton. And an atom that has only one proton, generally speaking, the hydrogen only has one electron. And hydrogen is special because hydrogen is, like I said, the first atom. And this first atom, its electron is in the first shell. And that first shell can only contain at most 2 electrons. The shells that come after that we're going to be discussing with the elements of interest, are going to be able to contain at most 8 electrons, but the first shell can only have at most 2. So the hydrogen atom with only 1 electron is missing an electron. It wants to fill its shell, and it's looking for a partner. Well, along comes oxygen. Now, oxygen has 6 electrons in its valence shell. And the 6 electrons are great, but oxygen very badly wants 2 electrons. In fact, oxygen is extraordinarily electromagnetic and goes over to the hydrogen and says, let's share. And hydrogen says, okay. And then oxygen hogs the electron. Similarly, another hydrogen comes along and says, I'm here, I'll share. And it shares with oxygen, but also shares unevenly. So oxygen gets the lion's share of the electrons. So if you have 8 electrons and you're down by 2 protons, it gives the oxygen part of this molecule a slightly negative charge. Whereas the hydrogen molecules, they don't have as much time with the electron, so they take on a slightly positive charge. So the water molecule has on the hydrogen side a slightly positive charge, and on the oxygen side a slightly negative charge. And as a result of this, other water molecules all seem to congregate in this organizational network such that they align, so that the hydrogen aligns with the oxygen and the oxygen aligns with the hydrogen, and these hydrogen bonds keep the cohesion of the water. And this is what gives water its characteristics. And incidentally, if you heat the water to the point of boiling, the hydrogen bonds are very, very short-lived, and you wind up liberating water into the atmosphere. And that's what happens. So hydrogen bonds are very, very important and are distinguished from other types of bonds. We just talked about oxygen and hydrogen sharing electrons. Whenever you're sharing electrons, even if it's unevenly, that's called a covalent bond. That's different than salt. Salt is sodium chloride, NaCl. What that does is that they bind together because one has a positive charge, one has a negative charge. So it's the charges, and that's called ionic bonding. Both covalent and ionic bonds are very, very strong. Hydrogen bonds are far more weak and can be broken much easier. But when you have a lot of them, it makes for great stability. Now, it turns out that hydrogen, oxygen, and carbon can all mix together to create something known as a sugar. We're familiar with glucose, which is C6H12O6, but that's not what we're talking about today. Today, we're going to talk about pentose, a 5-carbon sugar, and in particular, deoxyribose. Now, deoxyribose, in order to understand the structure of DNA, you really have to understand how we create what I'm going to call the sugar backbone. The sugar backbone, well, first of all, when we look at a sugar molecule, the way chemists go ahead and label the carbons is in a counterclockwise fashion. So we take the first carbon and label it carbon number 1. And then the next carbon 2, carbon 3, carbon 4, and carbon 5. That's how we label the carbons. And it turns out we'll start at carbon 5. Or really, you know something? Let's start at carbon 3 at the bottom. Carbon 3 actually is attached to something known as a hydroxyl group. And a hydroxyl group is nothing more than an oxygen and a hydrogen. And that hydroxyl group, in return, is covalently bound to a phosphate group. Now, that phosphate group is now bound to the carbon 5 position of the next sugar. And then that sugar's 3 position is bound to a hydroxyl, which is bound to a phosphate group, which is bound to the 5 position of the next sugar, and so on down the chain. And this is how we create a sugar backbone. And this is why we say it goes in a 5-to-3 orientation, from the 5 carbon to the 3 carbon to the 5 carbon to the 3 carbon, 5-to-3 orientation. Now, attached to these sugar molecules in the first position happens to be, well, when we talk about deoxyribose, we now need to talk about the nitrogenous bases. And there are 4 nitrogenous bases in DNA that are of interest to us. Those 4 bases are cytosine and guanine, thymine and adenine. Now, guanine and adenine both are molecules with 2 rings. 2-ringed nuclear bases are considered your purines. Pyrimidines have only 1 ring. That's your cytosine and your thymine. Now, it turns out we can make our lives a little bit easier. Instead of using the full words, instead of saying cytosine, we can just say C. And instead of saying guanine, we can just say G. And thymine, T, and adenine, A. It's just easier. It turns out that G and C have 3 hydrogen bonds. Yes, back to the hydrogen bonds. Hydrogen bonds are what hold the nitrogenous pairing together in DNA. And this is really important because whereas covalent bonds are really strong and a little bit less strong are the ionic, hydrogen bonds can be broken. And that's important because we need to be able to break those hydrogen bonds in order to be able to expose the substrate for either replication or translation, either one. So these hydrogen bonds are imperative to the structure of DNA themselves. It gives both strength to the structure and the flexibility for separation. Thymine or T and A only support 2 hydrogen bonds, whereas guanine and cytosine, C and G, support 3. This is important because often what we see in DNA are these islands of A's and T's, strings of A's and T's put together. This is important because there are fewer hydrogen bonds, and where there's fewer hydrogen bonds, that is a more ideal place for separating the DNA, unzipping it, and allowing something like helicase. Helicase is an enzyme which is used for unzipping DNA. And so it's easier to unzip the DNA where there, or at least to start the unzipping, where there are fewer hydrogen bonds. Now, one thing that you can do to help you remember that C and G have 3 bonds and A and T only have 2 bonds is that when you look at a C and a G, this is how I remember it. When you look at a C and a G, you can see that the C has looks a lot like the G, but the G has a little bar in it. So I say the C and G go together because they look the same, and the bar is the extra bond. And that means it has 3 bonds. So if once you know that, then the other is A and T, and that can only have 2 bonds. So you know that. Now we have to move over. Now, so we've just finished talking about one side of the DNA, because remember, DNA is double-stranded. So we talked about one strand, which went from the 5 to 3 orientation. Now the other strand goes in the other orientation, because in the same way that the bases were connected to the carbon 1, the bases are connected to carbon 1 in the second string. So all this does is cause the other string to go in the anti-parallel direction, in a 5 to 3 direction. So 5 to 3 is how we first described it. The second string goes in a 3 to 5 in the reverse orientation. And that's extremely important, because always in molecular biology, we talk about things like, for example, replication. And on the side, on the leading strand, the side that goes from 5 to 3, we have no problems. But on the lagging side, which we haven't explained yet, but that's the side which goes from 3 to 5 in the opposite direction, that has problems. And there's a special way where we go in fits and starts creating these things called Azaki fragments. And replication is a video for another day. And so is translation. But this is the structure of DNA. So essentially, the alphabet of DNA is adenosine, guanine, thymine, and adenine. And the way in which these bases sequence together will determine the way in which we can translate the code into ultimately some sort of product, potentially a protein product. But it goes through a series of steps, but this is where all of our genetic information is stored utilizing those 4 bases. So I hope you enjoyed this episode, and thank you for listening. And I can't wait. Next time is the origin of life. Bye-bye. Please like and subscribe. Sorry about that. Bye-bye.